Hello, my name is Anastasia Nickerson, and I'm a PhD candidate in the Department of Civil and Environmental Engineering. The title of my talk is Simulating Aerobic Biosparging of Aqueous Film-Forming Foam Impacted Soils. So first of all, PFAS, which are poly and perfluorinated alkyl substances, are a class of man-made environmental contaminants that are used in a wide variety of industrial processes and consumer products. For example, they can be found in Teflon pans, in fast food packaging, and in firefighting foams. So one common use of PFAS is in aqueous film-forming foams, or AFFF, which are a type of firefighting foam that's frequently used at military bases and airports to put out fuel fires. And because AFFF contains high concentrations of PFAS, these sites are often highly contaminated. PFAS are a broad class of compounds that come in many shapes and sizes. What they all have in common is that they have a lot of carbon-fluorine bonds, which are very strong and difficult to break down in nature. This is why PFAS stick around for a really long time in the environment. They can have short or long carbon chains, as well as different types of charged groups, such as the few examples that I have shown here. Sites that are contaminated with PFAS from AFFF often have other contaminants present as well. So for example, AFFF is often used to put out fuel fires, so fuel contaminants are often also present. One common example is BTEX, which stands for benzene, toluene, ethylene, and xylene. And remediation has often already taken place at these sites prior to knowledge of PFAS contamination. Since PFAS are more of an emerging contaminant, a lot of the time remediation such as biosparging has already occurred before they even find out about the PFAS that's there. Biosparging is often used for BTEX. It injects air or oxygen into the subsurface in order to encourage the microbial degradation of these compounds. And this obviously alters the aquifer redox conditions, and we believe this may inadvertently influence the microbial transformation of PFAS as well. So in my experiments, I'll specifically be using toluene as an example BTEX compound. Understanding these mechanisms on a laboratory scale should help us to understand what's happening at these sites where biosparging has already taken place or maybe is going to take place, so we can understand how it affects the release and maybe transformation of PFAS. So here I have a diagram of my experimental design. There are four AFFF impacted field collected soils that have been packed into columns, and two of these are receiving artificial groundwater that's sparged with oxygen. This is simulating our biosparging, where we have high dissolved oxygen flowing through. And then the other two are receiving artificial groundwater that is sparged with nitrogen, meaning it has very little dissolved oxygen. And there's also some toluene being pumped through the columns at the same time. And the column effluents are being analyzed for PFAS, for toluene, and for dissolved oxygen and pH. These columns were run between 2 and 4 months at a very slow flow rate. And one thing that I wanted to mention was that we did have unintentional stimulation of the anaerobic toluene degradation pathway. So even though our main goal was to stimulate toluene degradation via the aerobic oxic pathway, it can also be degraded anaerobically, which seems to have been happening in our columns. So all of the columns, both the oxic and anoxic columns, were microbially active. And this is a little image of what our setup actually looked like in real life. So a little bit messier, but you can see our 4 soil columns, our bottles of artificial groundwater. We have some syringes with the toluene that are being pumped in through a joint line into the columns, and then the effluent lines are being collected in those tubes for analysis. So now jumping into our results, here I'm showing concentrations versus time for these 3 particular PFAS, and I have their structures listed there. We have little ribbon plots where the blue ribbons are the range of the oxic column values and the red ribbons are the range of the anoxic column values. And what we kind of consistently see with a lot of PFAS is that there's this increase in concentration eluting from the oxic columns shortly after the start of biosparging. So it seems like the biosparging is helping to release these PFAS more quickly from the soil. So this is one of the major findings. We also wanted to look at the fraction eluted of the original soil PFAS mass. So essentially what we're doing is taking the sum of all the mass that's eluting in the water throughout, the 4-month experiment and dividing it by the original mass that was on the soil. So we want to see how much of that original mass has eluted, or maybe it's still hanging out on the soil. And when we compare the oxic and anoxic results in the two top panels, we don't see any drastic differences between these two for the top 10 highest concentration PFAS. However, when we compare them to the unaltered columns, which were in a previous round of experiments, these are not biologically active. In this case, we see a lot more eluting from the oxic and anoxic columns compared to the non-biologically active columns. In fact, it's between 120 and 800% more mass eluting in the biologically active ones than the non-biologically active ones. So this is important because it seems like the biological activity is somehow contributing to either release or transformation of PFAS, depending on the particular compound. So for compounds where the fraction eluted is greater than 1, that's a sign that perhaps we have transformation because we have more mass eluted than we originally started with. So jumping more into the transformation, we did a mass balance by summing up the mass eluted in the water with the mass that was left on the post-dilution soil, meaning that after the experiment was over, we took apart the columns and we analyzed the soil that was still inside, and we divided the sum by the mass in the original soil. So if this value is less than 1, that means that we have some mass unaccounted for, which perhaps has transformed. And if this fraction is greater than 1, that means that we have formed more mass of this particular compound. So certain types of structures seem to be more amenable to transformation. I'm not going to pronounce these acronyms, they're quite long and complicated, but as you can see in the top two structures that I have shown here, their structures are quite similar to one another. They have similar functional groups, and for both of these classes, there's a large amount of mass unaccounted for, meaning that we think they transformed. For the top class, there's only 0 to 1.4% of the mass accounted for, so the mass majority of it disappeared. In the bottom one, it was between 16 and 88%, depending on the chain length. So not only are the functional groups important, but the overall size of the molecule might be important as well. Meanwhile, other PFAS seem to have been formed. The most kind of drastic one was this class of perfluorinated sulfonamides, which had an up to 300% increase in total mass, meaning that up to 3 times the original mass was there by the end of the experiment. And our hypothesis is that maybe some of these more complicated looking structures, like the ones up top, perhaps these are transforming into these perfluorinated sulfonamides. If you take a look at those structures and imagine that you chop off the structure around that nitrogen, you essentially form what's on the bottom. So to conclude, we found that more PFAS mass eluted in the biologically active columns, both oxic and anoxic, compared to non-biologically active columns. We also found that select PFAS eluted at higher concentrations from the oxic columns than the anoxic columns immediately after the start of biosparging, and we saw additional mass of certain PFAS formed and missing mass of other PFAS, which points us towards transformation. The fact that the transformation was seen in both oxic and anoxic columns suggests that there may be more anaerobic pathways than was previously thought. Most of the transformation pathways that are in the literature are actually aerobic. To sum up, biosparging does affect PFAS release and transformation from AFFF impacted soils for particular compounds and classes. More research is needed to understand the particular mechanisms and why only certain PFAS are affected. Thank you.